Now that we've talked about the chemistry of life, we're going to start talking about the molecules that are then made of those elements that can combine and ultimately form all these important molecules that make up all life forms. When we talk about molecules, we can have two different categories. The first is inorganic, and inorganic are things that are non-living. So you can think of things like water or rocks, diamonds, silver, gases like oxygen and carbon dioxide. All of these things may be essential for life in the sense that we have to consume some of them or they're byproducts of life forms, but they themselves are not living. The compounds that are going to be a part of living organisms are going to be called organic compounds. Now, if you look at a cell, the cell is going to be made up of a lot of water, which is inorganic, but then the rest of it is going to be these carbon-based molecules, and we're going to talk about those today. We're going to talk about proteins, we're going to talk about nucleic acids, lipids, and carbohydrates. Now, all of those are going to be these carbon-based molecules that are going to help provide the building blocks for life. Carbon is very special because it has the ability to bind to 4 different atoms. We mentioned it last time we talked how it is going to have 6 electrons in its shells, and the outer outer shell is only going to have 4 electrons, and it has 4 spots that are empty. So those 4 spots can be filled with other atoms, and it has this ability to build these long skeletons or large complex skeletons that will build life forms. Here are some of the different forms that carbon, these skeletons can take. So we have, I want you to notice on each carbon, it's always going to have 4 bonds, and so we can count them. So right here it's bound to a hydrogen, here's a hydrogen, here's a hydrogen, another one. And all of these are covalent bonds. So carbon is going to bind with other atoms covalently. You can sometimes have double bonds. And so, for example, these 2 C's, these 2 carbons, are going to share 2 electrons with each other. But it still equals the 4 that it can bind with something. So here's 1, 2, 3, and 4. Carbons can make these really complex branched and unbranched structures, and they also can form rings. So we'll see some rings today when we talk about steroids and cholesterol. So here in this image, we can see the carbon atom. So the carbon, we're not showing any of the protons or neutrons. Remember, protons and neutrons are important and they're inside the nucleus, but they are not going to have anything to do with any of the actual binding with other atoms, okay? Because they're in the middle, they're kind of closed off. Only of the electrons are going to participate in any type of chemical bond. So if we look, we don't really have it drawn here, but there would be 4 empty spots on this carbon that it would be able to, it wants to fill with electrons, okay? And so it's going to be able to form 4 bonds with 4 other atoms, okay? So those 4 additional electrons will fill that outer shell. All of them are going to be covalent, like I mentioned before, which is a sharing of electrons. This is not a donation, this is a sharing. So they're going to be bound together, and this is a very strong bond. Okay. You can have lots of different carbon interactions, and they can form these very elaborate shapes. The proteins in our body, which are carbon-based, are going to have all these very complex and unique structures that allow them to do specific functions in our body. Structure is extremely important when it comes to how they function, and so we'll get to see some of those different structures today. Many of the times that we will look at carbon today, it will bind with hydrogen, oxygen, and nitrogen. Sometimes there'll be some other things, but the majority of the bonds are going to be with these 3 different elements. When you have molecules that contain only carbon and hydrogen, those are going to be called hydrocarbons. And the simplest is going to be something called methane. It's CH4. Now, when you're going to start seeing these these molecules written out like this. This is called the molecular formula. So it's telling you what atoms you have present. And then if it has a little number kind of as a subscript, that's telling you how many of those atoms you have. So for example, carbon, there's only 1 in methane. Technically, you could write a 1 right next to the carbon, but we don't usually write 1s. And then there's 4 hydrogens. So when you see these, that's what it's telling you. It's saying there are carbons and hydrogens, and there are 4 hydrogens in this case. So here is what methane looks like. So it's going to be a non-polar molecule. Remember, we talked about non-polar and polar, and so this is going to have even sharing of those electrons, so there's no positive or negative charges associated with the structure. Gasoline is C8H18, so it's going to be slightly more complex than methane, but similar in that it only has carbons and hydrogens. And the thing I want to point out is that we use gasoline to power our vehicles. Okay, and so within the bonds of these molecules, there is energy stored. And if you break those bonds, you can then perform work. For example, driving your car. So this is what the gasoline would look like. So you can count out how many of you have 8 carbons and 18 hydrogens. Fats are going to be made up of hydrocarbons. So when we talk about our fats, the main part of the fat is going to be these skeletons or these chains of carbons and hydrogens, and we'll talk about this when we talk about the fats and lipids. There are some oxygens in here, so this part is called the glycerol, and so this is not completely a hydrocarbon, but these chains are. And within these chains, there's just lots and lots of energy that we can break down and use to help our body. Okay, so these are very energy-rich molecules used by the body. The way that our body breaks down fats is very similar to the way that cars are going to break down gasoline. So when you're eating things that have fats in them, these are all examples of good fats. They will give you a lot of energy. If you eat too much of them, unfortunately, you then start storing fat. Now, the properties of molecules and compounds are going to depend on what's attached to the carbons. So the carbons are kind of the skeleton. They're the center parts, and then everything that kind of comes off of it is going to be then what determines how these things function. Now, it's really important to understand when we're talking about these molecules and function. So the way that something's shaped, it determines how it functions. So I want you to think about how you would use a spoon. So a spoon is shaped in a very specific way that it allows you to pick up liquid. Okay, we have different types of spoons. Some of them, we have slots in them so that you can actually drain liquid, but the shape is essential for the function. That's the same thing that happens with proteins. Okay, and proteins and other molecules is that they are going to have a very specific shape, and the way that their shape is going to determine how they function in the body or in any living organism. Okay, they also will determine how they'll be involved in different chemical reactions. For example, if you have a negative part of your molecule, for example, like water, if there's a negative aspect of it, when we do look at the water molecule, it's going to want to interact with positive things. And so that is showing you how the function is determined by that structure. Okay, and so we will look at a lot of these functional groups throughout the semester and see how they're interacting and how they work and basically provide the function for these different molecules. Okay. So here are a couple examples of functional groups. So we have right here, this is going to be called a hydroxyl. So if you have a hydroxyl on the end of a molecule, so this is an alcohol, for example, these are going to be able to interact with other molecules with this OH. And a lot of times what's going to happen with hydroxyls is it's going to join with another group that is going to create water as a byproduct, okay? Because you can see that there's already an H and an O, so we just need another H in order to make water. We have this group, which is called the carbonyl group. You You can find these in aldehydes and ketones. Carboxyl groups right here. This is the carboxyl. So it's got a C with a double bond O and an OH. And we have amino groups. These are going to be present in our amino acids that we'll talk about. And then phosphate groups. Phosphate groups form something called adenosine triphosphate, which is going to be the energy source that living organisms use. Now, these are, if you think about it, we're talking about chemistry right now. I will not ask you anything about an aldehyde or a ketone or anything like that, but you will need to recognize these different groups when you come into contact with them. And also, if there are certain groups like the amino group that's going to be associated with a specific biological molecule like an amino acid, which amino acids are going to be what makes up the proteins. And phosphate groups are important because they hold a lot of energy in their bonds that allow us to power our bodies. So when we look at how different molecules are represented, they can be represented in either a molecular formula way or a structural formula. Now, the molecular formula is what I've talked about a little bit earlier, where you have the number of each, or the type of atom, and then the number of each atom. So, for example, we have 6 carbons, 12 hydrogens, and 6 oxygens. Okay, this is called glucose, and we're going to talk a lot about glucose in this class. So it's just telling you the number of each one and what is present. When we talk about structural formulas, it's actually showing you how it is structured in space. So where's the OH pointing? Where is this group right here? Now, the thing I wanted to mention while we talk about this and why it's important to sometimes know the structural formula instead of just the molecular formula is that you can have 2 molecules, and these are both sugars. These sugars have identical numbers of carbons, hydrogens, and oxygens. However, they are not arranged in space the same way. So you're going to notice that glucose is going to be arranged like this, and we're specifically just looking at this carbon with a double bond with the oxygen and this OH group. And you can see that in fructose, it's arranged slightly differently. And you might say, well, why does it matter? Well, it matters a lot because the way that something's structured will depend on how it functions. And some of things like this where our bodies will only be able to break down certain types of structures based on what's, the chemical structure. So here, the situation when we have 2 different molecules that have the same, or I should say compounds, that have the same number of atoms of each type are called isomers. An isomer means that they're basically the same, but they have different structures. So they'll have the same molecular formula, but they'll have different structural formula. And it does matter what direction these things are facing. Okay, and we'll So we're now going to talk about these giant biological molecules that we build from smaller building blocks. So the things we're going to talk about are going to be carbohydrates, proteins, nucleic acids. Those are very large. We're also going to talk about lipids. These are called macromolecules. So macro means large, and these are very small in, kind of normal situations. But on a whole, if we look at the level of our body, they're actually quite large molecules. Now, I'm going to use a lot of different terms in this section, and it's going to be really important for you to be able to define these terms and understand what they're referring to, because it will be very confusing if you don't understand these terms. One of the first terms is going to be polymer. So a polymer is going to be a large molecule that's made out of smaller molecules called monomers. So monomer means one, polymer means many. So you can think of this as a necklace. So each bead is a monomer, and then when you string the beads together, they make this large necklace, which would be considered the polymer. So what we can see in this image right here is a kind of large molecule We have DNA on the outside, and then inside is going to be an enzyme, and we'll talk about what enzymes are. But this is going to be kind of a large molecule that's full of lots of little parts put together. Okay, so here are the 4 major macromolecules that we're going to talk about: lipids, proteins, nucleic acids, and carbohydrates. So it's going to be important for you to remember kind of what these different groups are, what they're made out of, and we'll go over all of that right now. So here is just a very simplified cartoon showing monomers going through a process called polymerization. So polymerization is a chemical reaction where you take individual pieces, these are little tiny units called monomers, and you string them together, like I said earlier, with a necklace, and you form them into something called a polymer. Again, mono means one, poly means many. So you go from one to many. Okay, and this whole process is called polymerization, where you're kind of joining small monomers together to form this larger polymer. Now, the way that polymerization takes place is a chemical reaction that I mentioned, and the reaction is called dehydration reaction. Now, this may sound like a weird word because you know what dehydration is. You don't have enough water, you're really thirsty. Well, dehydration does have to do with water. So when we are actually building these polymers, what happens is that we have a hydroxyl group, or an OH, on the end of one short polymer in this case, or it could be a monomer, and we have a hydrogen on another one. And when they come together, you have one H, two H's, and one O's, and those combine to form water. So when you actually bind these together, so we want to take this monomer and this monomer, and our monomer that's connected to a polymer, we want to make a bond between them. And so we get rid of water, which is dehydration, so you're like losing water, and in its place you'll have a bond. Okay, so that is going to be this first type of polymerization, where we're building kind of smaller polymers, adding on monomers, and creating a longer polymer. Okay, so dehydration reaction is a chemical reaction that removes a water molecule from the actual monomers that are involved in the reaction. Now, we can build molecules through the process of dehydration, but we then need to be able to break them down. So break the bonds that we form during dehydration. Now, the way we do this is the opposite, where we do something called hydrolysis. And when you break down the word hydrolysis, it means hydro, which is water, and lysis, which means to break. So you're breaking water, right? So in this process, we have a polymer, because a polymer, remember, has multiple of those small subunits. We then bring water in. Water then is going to break this bond, and we don't really need to know how it happens. We just need to know that water breaks it. And then we end up having 2 smaller polymer chains. And you can see that the parts of the water are now attached to those different chains. Okay, when we are eating and need to break down our food for energy, this is what's happening. So cells will need to break down bonds between those monomers, and they do this by the adding of water. You can also call this reverse dehydration. I prefer to call it hydrolysis or hydrolysis because it tells you in the name what's happening. Now, again, what I was just saying is that organisms are going to make macromolecules, but they also have to break them down. So when we eat food, that is a bunch of macromolecules. So we eat a hamburger, we are getting a lot of carbohydrates, we're getting protein, we are ingesting this and we're breaking it down. And the way that it's broken down is through that hydrolysis reaction. Okay, so we can then use the monomers that are kind of released from those polymers for building new molecules. Okay, so we're using our food for energy, but also for new building blocks. We're now going to start going into the 4 different groups of those macromolecules. The first groups that we're going to go over are going to be the carbohydrates. The carbohydrates are going to be the sugars. So carbohydrates are going to include both single sugars and polymers of sugars. And I'm going to give you some different names for how we talk about those. In animals, carbohydrates are going to be what we use for our primary source of energy. So it's what we eat, we then break it down, and we use it for energy. It also is going to provide the kind of building blocks or the monomers so that we can then build different organic compounds. Plants are going to be really important in the production of carbohydrates. They're going to be the initial source of where the carbohydrates are going to come from. We will eat them, we break them down, and then we can actually make one type of carbohydrate in our body. In this image right here, we have 4 different types of carbohydrates that we'll talk a little bit about. These are going to be polymers because they have many sugars strung together. The first is starch, and this is what you have when you eat a French fry. Okay, then we have something called glycogen. Glycogen is going to be something that animals make, a way that we store carbohydrates. And then we have something called cellulose, which is going to be what plants are made out of. And chitin is another type of polymer of carbohydrates, and that is what the shells and the body of the exoskeleton of insects and other groups like that are going to have. So we're going to start with the monomer, and we're going to call these monosaccharides. The word mono again means one, and saccharide means sugar. So if you, the word for sugar in Spanish is azúcar, and so you can kind of see how that word, where that comes from. Okay, so again, mono, single, and saccharide or sucar is going to be sugar. So these are going to be sugar molecules that cannot be broken down. So if you break it, well, you can break it down, but there is no sugar that's smaller than a monosaccharide. So you're basically, if you break a monosaccharide down, you're just going to end up with some carbons, hydrogens, and oxygens, but it won't be a sugar anymore. Okay. Some examples are going to be glucose. Glucose is going to be found in sports drinks, and glucose is going to be what we actually use for the energy of our bodies. We will be doing cellular respiration, and we break down glucose in that process. Fructose is going to be another monosaccharide, which is found in fruit. So when you're eating any type of fruit, you're most likely eating fructose. Fructose and glucose are those two that we saw earlier, where they both are going to be C6H12O6, but they have slightly different structures. And so you can see glucose right here, and here's fructose. To tell you the truth, there also is another isomer for this kind of sugar, which is called galactose. And galactose also is going to have the same number of carbons, hydrogens, and oxygens. We also will have deoxyribose and ribose. These are going to be sugars, and they're actually found in our DNA and RNA. Monosaccharides are going to be the main fuel that we use for cellular work. So again, when we start talking about photosynthesis and cellular respiration, the whole point of cellular respiration is to break down glucose. Glucose has a lot of energy stored in its bonds, and we will then be able to make something called ATP, which is what we use for direct work in the body. So here's glucose shown in 3 different ways. We have the structural formulas, so both of these show the structure. You can see the carbons, all 6 of the carbons, and then we have our hydroxyl groups and our hydrogens. This is what it looks like in a ring form. This is the linear form. And then this is a way that you can look at it without having to write all of the extra parts to it. I will not have you draw the glucose. You don't need to be able to recognize it in its different linear or ring structures, but you do want to know its importance, and that has, 6 carbons, 12 hydrogens, and 6 oxygens. The next step up, so when we have 2 sugars that come together, we have something called a disaccharide, and di means 2. So this is 2 sugars. Okay, so we can call this a double sugar. And the way that they are going to bind to each other is through that dehydration reaction. So in this case, we have a glucose and we have something called galactose. Each of these are going to have some functional groups on them that allow them to have a chemical reaction. There's an OH on the glucose, an H on the galactose. Those two things can form water. The water is lost, and in its place, we now have a disaccharide. In this case, we make something called lactose, which is what we have in kind of milk and other dairy products. So it's a milk sugar. And we'll talk a little bit about lactose, about how if you're lactose intolerant, you have a problem with being able to break this down. Okay, so lactose is an example of a disaccharide. Sucrose is also an example, and sucrose is going to be glucose plus fructose, which is table sugar. Now we're moving on to the polysaccharides. So remember, mono means one, di means two, poly means many. So this means that we are taking those small monomers of the sugars, those monosaccharides like glucose, and we are stringing thousands of them together in order to make a polysaccharide. So these are quite large. They can be very complex and they can have lots of different structures. For example, what we're looking at right here is a glycogen. And so each of these individual little things is a sugar. And so they are structured in this very complex branched formation. Now, polysaccharides are going to tend to be either structural or storage. So we're either storing a bunch of energy in the form of these long chains of glucose, or they are going to help for structure of, say, a plant. Okay, the three main types that we're going to talk about are going to be starch, glycogen, and cellulose. And you will need to know the difference between these three types. Okay, one main thing I wanted to point out is that they look very different structurally. So depending on how they bind to each other, they are going to have very different structures, and that is going to then cause how they function to be different. Now, they all are going to be bound by dehydration reactions, but they are not necessarily going to be kind of in the same way that they're bound. So we still have to use that dehydration, but there can be slight differences in how the binding takes place. So we'll look at starch, which is what we would find inside of a potato. Glycogen is something that we find a lot in mussels. It's the source of energy that mussels use first. And then we have cellulose down here, which is going to be a structural component of plants. So starch is a storage polysaccharide. It's going to be long strands of the glucose monomer, and this is what plants use for energy. Now, the plants are going to perform something called photosynthesis, where they make glucose, and they want to store the glucose in these long chains that they can then store, and this is called starch. Now, plants make it for themselves so that they can use it when they need it. So, for example, a potato is produced for the plant to store a bunch of energy for when the plant will need it. However, animals like us will take advantage of that storage crop of starch and use that to eat it. So animals can digest starch through hydrolysis, and we get the energy from it. Our next polysaccharide is glycogen, and glycogen is very similar to starch, but it's the way that animals store their excess glucose. Okay, so this is the storage polysaccharide for animals. Anytime you have excess sugar, which is a problem for a lot of humans, is we get lots of sugar, we will then need to store it because we can't have it just sitting in our body circulating in our blood. If we have it there, we start to have issues like diabetes. So we need to have a way to store excess sugar. And we do this by forming these long polysaccharides of the sugar. Now, unlike starch, it's going to have a slightly different structure. It is branched, and so you can kind of see the branch structure in this image right here. So not just these long chains going all the same direction, but lots of kind of branch structures. And the glycogen will be stored in mainly two locations inside the liver. And so this is all the pink that you see here is glycogen granules that are in liver cells. But we also store a lot in our muscle cells. So muscles are going to be one of the locations that have to use a lot of energy. So think about writing the notes right now. Your hand is having to use many muscles in your hand and your forearm, and all those muscles are going to require energy to be able to contract and move. So one of the places that we have a lot of stored glycogen is in the muscles themselves, because they're going to need sources of glucose. Now, this is the reason why athletes like to do something called carbo loading before an event. So if you know you're going to run a marathon or do some very strenuous activity, it's important to carbo load, and so have a lot of excess carbohydrates in your body so that you can store it and your muscles will have plenty of energy. If you starve yourself before a huge event, you are not going to last very long, because you're already depleting your sources of sugar, and so that's why you do this carbo load event. And this is one of my favorite carbo load events. I wouldn't suggest ever carbo loading the day of a race, especially fettuccine alfredo. That will not end well. Our last main polysaccharide is going to be cellulose, and cellulose is the most abundant organic molecule on the planet. Every single plant that we have is made out of cellulose. So cellulose is going to be the structural fiber that gives the plants the ability to stand upright. They form cables, and we consider them fibers or fibrils, which is just kind of a smaller fiber. And these are going to be found in the cell walls of plant cells, and we'll talk about cell walls when we get to the cells. Wood is also going to be made out of cellulose. It's a little bit of a tougher type of cellulose, but this is going to be very strong, and it forms these kind of long chains, not as branched as we saw in the glycogen or the starch. Now, it turns out that the structure and the rigidity that cellulose provides for plants is going to cause issues for humans and other animals trying to digest it. So cellulose has a very specific structure that has these linkages that our bodies cannot break down. So when we have starch and glycogen, our bodies can easily break these down. We have the right protein enzymes. They're going to be able to chop them up and do that hydrolysis reaction. However, the way that cellulose is structured and how it's bound to each other makes it so that our bodies cannot break down cellulose. Okay, so if you eat a bunch of grass, you are not going to be able to break down it, and it's going to come right out of you. Now, it's important to eat things that have cellulose. So spinach is made out of mostly cellulose, grapes, the skin, and broccoli. So we eat those things, and they're all going to be called insoluble fiber, which means that we can't technically digest them and extract the glucose from them, but they are important for cleaning our digestive system. So it is important to still eat these. Now, humans can't break down, but other animals can't break down either. So, for example, cows or deer, these animals that eat almost exclusively grass, they also, on their own, would not be able to break down cellulose. However, they have this very interesting relationship with microorganisms that live inside their guts that allow them to break down cellulose. So the digestive system of a cow is very complex. It has multiple stomachs, and it also has some special things that it does. For example, it will regurgitate some of the plant matter and have it come back to the mouth, and they'll re-chew it. So they have to really break down the cellulose or the plants until it's very small. And then once it gets into the stomach, there are going to be microorganisms that are going to help break down the cellulose. Now, this is called a symbiosis, where the two organisms, so the cow and the microorganisms, are actually going to be working together. The microorganisms get to live in a nice, humid environment inside of the cow, and the cow gets to have cellulose broken down into glucose so that it can use it for energy. There are lots of different types of mutual or symbiotic relationships like this. For example, termites will also have little tiny microorganisms inside of them that break down wood, and that they have to live together, otherwise they won't actually survive. One thing I like to mention with this is, how do babies, for example, baby calves, how do they get inoculated with the microorganisms? And this is going to sound kind of gross, but oftentimes the baby animals will have to eat some of the poop of the mother, and that will actually get the microorganisms into their guts. It's kind of gross, but it is an important stage of life. Okay. And again, the undigested fiber keeps the digestive system very healthy. So if you're really constipated, sometimes they'll say you need to have more insoluble fiber. All right, so we want to talk just a little bit more about the properties of sugars. So sugars are what we call hydrophilic. We've seen this word again, hydro. So this part of the word means water. This part of the word may not be something that you've heard of before, but philic is going to mean love. If you know what Philadelphia, so that is the city of brotherly love. So phila means love. So sugars love water. And my best example of this is, think about when you've ever made Jell-O. So when you make Jell-O, you have these sugar little granules, you put them in water, you stir it, and it completely dissolves so that at some point you have a homogeneous mixture where everything is completely mixed well. Now, some molecules will not do that. So they're going to want to stay separate from water. They fear water. But sugar is not one of those. So sugar is able to dissolve in water, like we see in this image. All right, so now we're going to switch over to lipids. And lipids are actually going to be the opposite of sugars in the sense that they fear water. If you've ever made a salad dressing, or if you have salad dressing that you bought from the store, you're going to notice that when you let it sit, specifically the oil and vinegar types, it's going to separate out where the fat is in one spot and kind of the vinaigrette and all of the seasonings is at another place. The reason why it does that is because lipids and fats are something that we consider hydrophobic. And phobic in this case means fear. So they're going to fear the water. So the lipids are going to include a lot of different types of fats. We also have things called steroids, and we have cholesterol. So we're going to go over these fats now. So they are hydrophobic, fearing the water. And so they like to separate themselves from water. Now, lipids are this one group that are slightly different from the other macromolecules that we're going to talk about. So in each case, when we talk about the carbohydrates, the proteins, and the nucleic acids, we're going to have a defined monomer, or the small building block, and a defined polymer. Lipids don't have that. They do not have what we would consider a monomer in the same situation. So I will make a list kind of comparing all of these together, and you'll see that this one is slightly different from the other three groups. Our first category of lipids are going to be the fats. So fats are going to consist of something called glycerol, which is then going to be joined with 3 fatty acid molecules. And the 3 fatty acid molecules would be joined to that glycerol through a dehydration reaction. So that is the same, that's similar throughout all of the macromolecules. They're all going to use that dehydration reaction. Now, fatty acids are going to be these long hydrocarbon tails that are going to have a lot of energy within them. So they're going to be mainly just carbons and hydrogens for, in some cases 30 to 40 carbons strung together, and then each carbon is going to have some hydrogens on it. These are very energy efficient, and so often when you're trying to eat healthy, they do suggest you eat healthy fats. For example, fish fats or nut fats. All of those are going to be very energy efficient and allow you to have energy. So here is what we are talking about with the structure of these fats. So they're going to have this orange structure right here, which this is called the glycerol. So this is the glycerol, and it is going to then be attached to up to 3 fatty acid tails. So here's a fatty acid tail. These are all fatty acid tails. I'm going to talk to you why this is bent right here, but they can either be straight or bent. The way that the fatty acid tail joins to the glycerol molecule is that it does a dehydration reaction. So you have the H and the OH, that then makes water. So right here, this would be considered a triglyceride because it has 3 of the fatty acid tails. Now there are 2 categories of fats. we have unsaturated fats and saturated fats. The big difference between these two types is the number of hydrogens that are attached to their hydrocarbon tail. Now what you're going to notice right here is that we have kind of this carbon skeleton, and then we have these hydrogens on each side. But we get to this set of carbons right here, and we see that instead of having, there could be an H right here and an H right here, but instead they're bound to each other with a double bond. Now this double bond causes a bend in the shape, so it's no longer a straight line. Now that bend is going to make this behave very differently. So remember, structure and function. So how it's structured will determine how it functions. So it has the less than the maximum amount of hydrogens on its carbon tail. Okay? And this is going to be found in plants and fish fats mostly. They are usually going to be very high in unsaturated fatty acids, and unsaturated fatty acids tend to be better for you. Now the interesting thing, so this is where we get that structure function, is that because of that bend, when we have unsaturated fats at room temperature, so that's about 70 degrees, these will be liquid. And the reason why they're liquid is because they cannot stack on top of each other because of this little bend in that tail. Okay, so they'll be liquid at room temperature. So when you're thinking of unsaturated fats, think olive oil or any of those vegetable oils, things that are liquid at room temperature. So plant oils are a good example of this. Okay, so the bent shape in the tails make it so they cannot line up. You could take an unsaturated fat and lower the temperature enough that it probably would start to freeze and be solid, but at room temperature it will be liquid. Now on the other side we have saturated fats. So saturated fats are going to be things that are found in all the most delicious foods, in my opinion. Bacon and meat, sausage, butter, cream, all of these things that are very fatty, okay? And they are going to actually have a straight shape. So what the big difference between saturated and unsaturated fats is, you're going to notice that there is no double bond in this chain of carbons. So these are fully saturated, which means that they have the fullest amount or the highest amount of hydrogens possible for this chain. Okay, the unsaturated fatty acids could have potentially had 2 more hydrogens, but instead it had a double bond. In saturated fats, you have only these hydrogens. There are no double bonds. This makes it a very straight structure, which means that when it lines up at room temperature, it's going to be solid. Okay? Most animal fats are going to be considered saturated fatty acids. There are some that are unsaturated, but most of them are going to tend to be fatty saturated. They're solid at room temperature. So think about this. When you make some bacon and you let your bacon grease sit in the pan, it forms a solid pretty quickly. Okay? Butter, great example of a saturated fat. Those things are going to be solid at room temperature. You can heat them up and they can become liquid, but you have to put in that heat. Okay, so the linear shape, the very straight shape of their tails allows them to stack on top of each other very regularly and causes them to have that really solid structure. Now, I told you that unsaturated fats tend to be healthy and good for you. Saturated fats, eating them moderately will be fine, but you can actually start to cause health issues if you have really high levels of saturated fats. So if you're eating a pack of bacon a day and using lots of butter and heavy cream and eating tons of sausages and red meat, you're going to probably have some issues with cardiovascular disease. Now, you can have a mixture of saturated and unsaturated fats together. So not everything is just going to be purely saturated, something's going to be purely unsaturated. For example, chocolate has both saturated and unsaturated fats because it has cocoa butter in it. So, but it also, some fats are going to have that, milk chocolate's going to have milk, which is going to tend to have those saturated. Now, when you have this chocolate at room temperature, it's going to be usually solid. But the second you raise the temperature just a little bit, so you put a piece of chocolate in your mouth or even in your hand, if your hand gets hot enough, it is going to start to melt. Just a little more about the healthy fats. So some fats are actually good for you and essential. Omega-3 fatty acids are going to be something that a lot of people take supplements for. These are going to be found naturally in fish oils and nuts, and they actually can reduce your risk of heart disease. So if you have a balanced diet of both unsaturated fats and saturated fats, you can still enjoy having bacon every once in a while, but you also are going to have enough of this unsaturated that can help make sure that your heart is healthy. Steroids are going to be our next group of lipids. They're going to look very different from our fatty acids. You can see that right here, this is cholesterol. So cholesterol is a steroid, and it's going to have these rings. So it does not have the same linear structure of those fatty acid chains. Because of this structural change, it's going to function in a much different capacity. So steroids are going to be important in really important functions within the body. Okay. All steroids will have a very similar structure with these carbon rings. We call these fused carbon rings because they're attached to each other. And all of the steroids that we're going to talk about, including testosterone and estrogen, are all going to come from, first, cholesterol. So cholesterol is the starting product, and then you can modify it by taking off some of these extra groups and turn it into testosterone and estrogen, which are sex hormones. Okay. Depending on what is attached to the chain, you will then have different functional groups, which will mean that they'll function in different ways. Let's start with talking about cholesterol. Cholesterol is an essential molecule for the body. It's going to be a key component that's inside of our cell membranes. And so you can see these little cholesterol molecules that are present within the cell membrane. We haven't talked about the cell membrane before, but we will in a little bit. But this is going to be the membrane around every single cell. Okay, and so the cholesterol is going to be inside of the membrane that helps to keep it fluid. Now, each steroid that we are going to talk about, and we're not really going to go into depth too much about the steroids, but all of them are going to start with cholesterol. So you have to have cholesterol as a starting part, the starting structure, and then it can be modified and altered to form the different steroids for the body. So you do not need to know this chart, but I just wanted to show it to kind of explain how cholesterol can be turned into different steroids. So this is what cholesterol looks like, and this one looks more complicated because it has all of its carbons numbered. So you can see what these numbers, they just represent where carbons are. But cholesterol can turn into both androgens. So androgens we tend to think of as male sex hormones, although the amount that males have is higher, but females also will have androgens. They're essential for female behavior as well. We can also make estrogens, which we tend to think of as female hormones, but men will also have hormones, female hormones as well, estrogen. It's more of a balance, but how much of each one you have that determines your biological sex. We can then make other things like cortisone, aldosterone, glucocorticoids. All of these are important steroids for the body. So this image shows a cholesterol molecule and then testosterone and a type of estrogen. So you're going to notice that they have largely very similar structures, but those functional groups are very different from each other. Specifically, actually, it's only two parts of the chemical structure that are different, but that structure makes it function in a completely different way, which then causes male characteristics like this in this lion with a huge mane, and then on females, they do not have a mane. So both behavior and physical appearance can be altered by the presence or absence of testosterone and estrogen. While we're on the topic of steroids, I wanted to mention a little bit about anabolic steroids, and specifically anabolic testosterone. Now, anabolic testosterone is going to be synthetically made, which means it's made in the laboratory and not within the body. And it's going to mimic what naturally occurring testosterone does in males. So in males, testosterone is going to be released at very high levels during puberty, which allows for bone and muscle to grow. Okay, so you often will see a very dramatic height or growth spurt for males going through puberty. They often can start, very short during middle school, and then by the end of high school have grown a foot. And they also are going to increase muscle mass. Testosterone then is continuously produced throughout the life of the male, and that continues to maintain those different masculine traits. Now, anabolic steroids are going to mimic these actions, and anabolic steroids were created to help certain individuals. So initially, they were used for individuals with cancer or AIDS that were having issues maintaining body mass and kind of their muscle and bone. It also now is used for gender-affirming hormone therapy. So if you have female to male transitioning, you can have testosterone. There also is going to be the opposite, where you have male to female transitioning, which you would use estrogen. So it doesn't have to be testosterone. But that is something that doctors are kind of helping patients with. However, athletes have also used anabolic steroids to help build up muscle quickly. And it's illegal in professional sports and in collegiate sports as well, because it is considered cheating. Now, there are lots of side effects with taking anabolic steroids, which is why you have to be under a doctor's supervision for specific types of hormone therapy. But a lot of people take it and buy it illegally, and they can have side effects if they give themselves too much. So some of the side effects are roid rage, depression, liver damage, shrunken testicles, reduced sex drive, and infertility. The last three are interesting because you'd be, well, that's weird, because those seem like if you have a bunch of testosterone, why would you have shrunken testicles? Well, what happens is your body is constantly monitoring the different levels of hormones in your body. And if all of a sudden you're getting excess testosterone, it's going to think it's making too much. And so your testosterone producing testicles will shut down and not produce as much testosterone, and in fact shrink. Okay? And so if you don't have the real testosterone circulating in your body, and that's going to affect your sex drive and potentially can cause infertility. You can also have horrible acne that can be caused by anabolic steroids. So I don't suggest taking anabolic steroids for purely muscle mass. If you are taking them, please be under a doctor's supervision, because it can cause some very bad side effects. We're now going to move on to proteins, and proteins are amazing. They basically do and make up everything inside the body. We're going to talk about structural proteins, storage proteins, contractile proteins, transport proteins, enzymes. All of these are doing and making everything in your body. Okay, so we're talking about things from your hair to your blood cells, your muscles. All of that is going to help the body stay alive and keep on functioning. So proteins are going to be made of amino acids, which are going to be the monomer. So amino acids are going to be the small units, and we'll talk about what the amino acids are. And if you build a larger chain of these amino acids, you build something called a polymer or a polypeptide, which I'll mention in a little bit, which is a protein. These are going to be the most elaborate and the most diverse molecules of life. And people study their entire lives specific proteins to try to understand how they are functioning in the body. Many diseases and syndromes result from non-functional proteins. And so if we can understand how these proteins are made and how they function, we ultimately can cure a lot of diseases. So this is a very important area of biology. They are going to be instrumental in almost every single aspect of the cell and in our bodies, allowing us to function. These two images right here are an immunoglobulin, which is going to be important in the immune system. So the immune system is going to have lots of proteins that are associated with it that are able to recognize foreign molecules like viruses or bacteria or foreign organisms and attack them and get rid of them so that we can defeat it. So as I mentioned, amino acids are going to be the monomers. We have 20 amino acids in the body, and these amino acids are all going to have a specific name. You do not need to learn all the names of all 20 amino acids. We'll talk about what you'll need to know for amino acids in general. But you can see you would string all these different amino acids together, and they will create something called a polypeptide chain. Now, the reason why we call it a polypeptide chain and not just a polymer chain is that when we have the bonds between the amino acids, we call those peptide bonds. Peptide is just another word for protein. So they are protein bonds, basically. So each of these 20 amino acids will be strung up, and every protein is going to have a unique sequence of those amino acids. Now, I just want to mention that having the wrong amino acid can completely destroy the function of proteins. We'll talk about sickle cell anemia, which is one amino acid that's been changed in a protein that causes kind of a disease in individuals. When we look at the amino acid itself, so these are on the previous page, they look like little beads, but if we zoom in and look at the amino acid, all amino acids have kind of the same structure. They're going to have what we call an amino group, we have a carboxyl group, we have an H, hydrogen, and we have this side group. Now, this side group is what is going to be the functional group. So each amino acid is going to have a unique side group. That side group is going to determine how that amino acid is going to interact with other chemicals and molecules. Okay, so this side group is going to be really important in determining how that amino acid is going to function. So this is just reiterating what I just said, but that fourth group, that R group or that side chain group, is going to be determining what that amino acid is going to do. Okay, so it's going to have its special characteristics. For example, this is the amino acid leucine. So you're going to notice that the amino group is there, we have our hydrogen, we have our carboxyl group, and this structure right here is going to be called a hydrophobic side chain. Hydrophobic meaning fearing water, so it's not going to want to interact with water. Okay, and so that is going to determine its special characteristics. This other one is called serine, and this is a hydrophilic side chain. So these two side chains are not going to want to be next to each other because one's hydrophobic, one's hydrophilic, so they will not want to interact. But hydrophilic side chains will want to interact with other hydrophilic side chains. For example, you can imagine that serine would not have a problem with being surrounded by water or being around sugars because all of those would be hydrophilic. This diagram shows the different types of side chains. So we have some amino acids that have side chains that are going to be nonpolar, which means that there's no positive or negative charge associated with these. Okay, so these are all nonpolar. Other side chains will be polar, so they're going to tend to have a charge differential on their amino acid. Okay, we'll have some that have a positive charge and some that have a negative charge. And all of these, again, are causing these to behave in a very specific way. So they'll only interact with specific things. For example, positive charges are going to want to interact with negative charges, and negative charges are going to want to interact with negative charges. But you would not have two negative charges interacting with each other because they would actually repel from one another. When we form the polymers of these proteins or polypeptides, we still use the dehydration reaction. But again, I mentioned earlier that because we're dealing with amino acids, we call these peptide bonds. But the peptide bond is formed through the dehydration reaction, like we mentioned before. Polypeptides are these long chains of amino acids. You can kind of think of the polypeptide as the stage right before we get to what we consider the protein. The protein is the fully functional end product of this whole process. When we have the polypeptide, it hasn't quite got all of the folding and kind of structural organization that a protein has. And I'll explain all that structural organization in a second. And our bodies have tens of thousands of proteins. People actually work, there's scientists that are trying to kind of identify all the proteins that we have in our bodies. Because again, you could potentially start to cure diseases if we know, well, this disease has to do with a non-functional protein. If we know where that protein is and what it does, we could then potentially fix it. So here is the dehydration reaction, just to kind of reiterate. So you would have your amino acid group, you would have one of your carboxyl groups interacting with one of the amino groups. We have water here, so water is lost in the dehydration reaction, and in its place we are left with a peptide bond. Okay, and so that would be that polymerization. We are doing the dehydration reaction. So here is an example of a long polypeptide. So we have all the different amino acids strung together. So this again would be a polypeptide. It's not quite what we would call a protein yet. So how do we have so much variety? Okay, so those 20 amino acids can be strung in any different combination. You can kind of think of the amino acids as letters. So each letter can be arranged in different combinations to make new words. We're constantly making new words. I feel like young people are constantly making new words that I don't know what mean, and I have to Google and go to Urban Dictionary to figure out what they mean. But we have this endless possibility of the different orders. For example, I was talking about sickle cell anemia earlier. So this is a normal sequence for hemoglobin. Hemoglobin is an important protein that's involved in red blood cell formation. And so we have this it's 146 amino acids long. Here is a second chain polypeptide. However, this is a sickle cell hemoglobin. And you're going to notice that there is one amino acid that's in red right here, and this is a valine. Right here, it's glutamine. I can't remember all of the three-letter codes, but you're going to notice that it's not the same amino acid. Because of that, the structure is completely different from the kind of the normal situation. So this is a sickle cell polypeptide. This is the normal polypeptide. If we look at the total structure of the red blood cell, this is a normal red blood cell, and this is the sickle cell red blood cell. And so having that one change, that structure will completely alter. And people with sickle cell anemia are going to have kind of intensive care throughout their entire lives to make sure that they are not suffering the effects of sickle cell. And we'll talk about sickle cell a couple more times because it's an interesting disease, because it actually has some benefits if you live in certain areas of the globe. Okay, when we talk about structure of proteins, we talk about different levels of the structure. And this first structure is called the primary structure, which is the sequence of the amino acids. So if you just go through and you could see what sequence they're in. Having one change, and sometimes you're going to have more than one change, that will then have a dramatic impact on the actual protein. Usually a change is going to, specifically if you change which amino acid is present, you're definitely going to change the structure and therefore the function of that protein. So here is a little bit about protein structure. So to go from a polypeptide to a protein, you have to have structural organization take place prior to being a functional protein. So protein is kind of like our end game. The polypeptide is this intermediate where it still needs to have a little bit of structure take place. So functional protein can often have more than one polypeptide chain put together, but it also, even if it is just one chain, it's going to have to be folded and twisted and coiled into a very unique shape, because shape is essential for function. So the way that it's shaped is going to determine how it functions. You can kind of think of it as you have a key and it has to be in the shape of the correct key, otherwise it will not be able to fit the lock. And so we're going to have this structural importance and this unique shape. So there are 4 levels of organization. We're going to go primary, secondary, tertiary, and quaternary, and we'll talk about those 4 different levels. So let's look at the structural levels a little bit closer. So our first level of organization and structure is called the primary structure. And again, that is going to be just the sequence of the amino acids. So what order are they actually in? The second step is called secondary structure, and there are 2 different forms that this can take. You can either have something called a beta pleated sheet, or an alpha helix. Okay, and these two ways are dealing mostly with hydrogen bonds between hydrogens and oxygens. Okay, and so these are going to be those interactions, and you can sometimes get these coils or these beta pleated sheets. So they're kind of a fold. Those then are going to fold even more, and you can have both beta pleated sheets and alpha helixes all put together. And so there's more folding and more interactions between usually the side groups at this point. So those R groups that we talked about that are unique, those then are going to start to interact with each other. Now, you may stop here, it's possible, but there are many proteins that are actually going to consist of multiple polypeptide chains that have been folded. So you can imagine that potentially there could be 4 of these tertiary structure. Those then will all hook up with each other and they'll make this quaternary structure, which is a polypeptide or a protein with 4 polypeptides. So it's kind of hard to see them, but there would be 1, 2, it looks like 3, and then 4 back here. So there are 4 polypeptides that have all been come together, and so that would be considered a quaternary structure. And just a reminder, so primary is 1, secondary 2, tertiary is like a 3rd structure, and quaternary means the 4th level of structure. Now the 3-dimensional shape of the proteins are essential. So we see, on here we can see different structural organizations. And the structure will be essential for the function, and if it's not shaped correctly, it will not function correctly. And non-functional proteins are going to be very dangerous, okay, and they can cause a lot of health issues. Right? The ability of a protein to have the right shape and be able to recognize other molecules and bind to other molecules is essential for its function. And so that structure, again, I can't stress this enough. Structure and function are intimately tied. If you're someone that's thinking about going into anatomy physiology, we talk about the same thing, how the structure of the body, the bones, the muscles will determine how they function. So we're on a molecule level, and that's how it works. So things, the way that they're structured is going to determine how they function. So here are some different proteins that we have, and most of them are labeled. For example, this one is called insulin, which is really important in blood glucose regulation in the body. We have trypsin. This is ATP synthase, which we're going to talk a lot about when we go through the cellular respiration material. This is ribisco, which is important in photosynthesis. We have alcohol dehydrogenase, which I think breaks down alcohol. And this is an antibody. Okay, so this would be able to recognize different foreign objects in your body and kind of mark them for destruction. And this is hemoglobin, so hemoglobin that we would use for carrying oxygen throughout our blood. Okay, so again, function is going to follow form, or function and structure and function are extremely tied to each other. If a protein is not shaped correctly, it will not function correctly. And this is just an example of how the muscle, skeletal muscle, is arranged. And you're going to notice that it's in a very highly structured pattern, and that is all essential for how muscles contract. And so if the muscle is not shaped correctly, the cells down on that molecular level, those will not function. When you have proteins that are misfolded, they can cause different diseases. And here we have kind of example of a normal protein, and these are on the brain, and these are what we call prions. And you may have heard of prions when we were talking about them in lab with mad cow disease. So prions are going to be misfolded proteins. Unfortunately, your body cannot break them down. So they do not have the right machinery to break down a prion. So unfortunately, prions start to get clumped up in your brain, and they eventually will cause some pretty bad nervous disorders. For example, Alzheimer's, Parkinson's, and mad cow disease are all examples of nervous system disorders that deal with misfolded proteins. Okay, so again, right there, the structure of it is essential. If we do not have the right type of enzyme or protein that can break down these misfolded proteins, they will, in essence, kill you. Okay, so you have to have the right machinery, and everything has to be folded correctly and have the right structure. Now we're going to transition to nucleic acids, and this is going to the reason why I want to make this transition between proteins and nucleic acids is because you might say, well, what actually determines the sequence of amino acids and how it folds in your body? Well, it's going to be the genes, and the genes are going to lead us to our next major group of macromolecules, which are the nucleic acids. So nucleic acids are going to be DNA and RNA, and they are going to hold the instructions. Specifically, the DNA holds the instructions for all the proteins in your body, and we will actually go through the process of how this happens. But nucleic acids are going to come in the form of DNA or RNA. DNA is going to be kind of the blueprint or the very important instructions, and RNA is going to be a copy of those instructions. And so it's a little bit, you can kind of send this in different places because it's not as important to maintain. Okay, so we'll talk about how this process happens in future lectures. So nucleic acids are going to be the macromolecule that store information and have all the instructions for building everything in the body. Okay, they're going to determine what color your eyes are, if your hair is curly or straight. It's going to determine the color of your skin. It's going to determine everything about you. Maybe not everything. So there are certain things like behavior that some of them are built in the genes, but basically they're going to provide all of the instructions. There are two types that I mentioned. We have DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. Now, both of these you're going to want to know their full names just to be able to recognize them. And they are going to be very similar in some ways and have some differences in others that are extremely important. So if you can see in just this little image right here, this is DNA, and DNA is going to have 2 strands, while our RNA is only going to have 1. Okay, DNA is going to reside inside of the cell in a specific location called the nucleus. Now we've heard of a nucleus before, which was with the atom. So inside the nucleus we had protons and neutrons. The way that the nucleus is similar in this case is that when we have our cell, we're going to have a center location where all the DNA is going to be stored. And this is going to be a really important and kind of very locked down part of the cell because we want to make sure that DNA is not messed with. Inside of the nucleus, you're going to find the DNA that's found in these structures called chromosomes. Okay, and so DNA is going to be tightly wrapped around itself and form this chromosome. And we'll go over more about the chromosome structure later on. A gene is going to be a very specific location on the DNA. So it'll be a sequence, and we'll go over what these letters mean. We have A, G, C, T. Those are going to encode a very specific sequence that is going to then make a polypeptide. So we're going to be able to take this code that the DNA gives us, turn that into amino acids, which then is going to give us a protein. The code is in nucleic acid language, which has these different things we're going to call nucleotides, which I'll explain. But that language can then be translated into a different language, which is the amino acid language. So nucleic acids are the polymer. So they are the large structure. But the polymer is made out of monomers, and in this case, the monomer is called a nucleotide. Okay, I would definitely make note cards with these different terms on them because they can get confusing. A nucleotide is the monomer for the nucleic acids. A nucleotide includes 3 things. It's going to include a nitrogenous base. This means it has a nitrogen in it. So if you're going to notice, you look in here, there's an N. That's nitrogen. It's going to have a sugar, and it's going to have a phosphate. So every nucleotide has these 3 things. So that is our small monomer for the nucleic acids. Okay, so 5-carbon sugar, which is either going to be deoxyribose or ribose. We're going to have a phosphate group, and we're going to have a nitrogen-containing base or a nitrogenous base. And there are 4 different versions of those. We have one that's we consider A, we have a G, we have a C, and we have a T. And I'll give you the full name of those. The way that you bind nucleotides together is going to be, you guessed it, a dehydration reaction. So those are going to link those different nucleotides together, which ultimately is going to form our nucleic acid of either a DNA or an RNA. So we mentioned that in the nucleotide, you have to have a sugar, and there are two different types of sugars. There are both 5 carbon sugars, which means that you can count the carbons. So we have 1, 2, 3, 4, 5. This one also has 1, 2, 3, 4, 5. Now, there are 2 versions. We have something called deoxyribose and ribose, and you're going to notice that this red OH and this red H over here are where the difference between these two are. So the deoxyribose means it's deoxy, so think about it as like it has 1 less oxygen on there. So this is going to be the sugar that's used for DNA, and this is going to be the sugar that's used for RNA. So that's where that deoxyribose and ribose for those 2 nucleic acids comes from. So let's look a little closer at the DNA. So the DNA is going to have what we call a double helix. So it's going to have 2 strands that are going to wrap around each other, and in the middle we have them bound to each other through the nucleotides. Okay, and we'll start talking about those nucleotides. The bases are bonded to each other with hydrogen bonds. So within the actual nucleotide there are covalent bonds, but what binds these bases together, which are these nucleotides, they are going to be hydrogen bonds. So they're very easy to break, but there's a lot of them, so we have a strength in numbers. Okay, so again right here. This allows for unzipping of the DNA and will be extremely important in how this functions, and allows us to open it and make copies of it and then close it again. Now we have 4 of the nitrogenous bases. There are cytosine, guanine, adenine, and thymine. You do not need to know the actual structure of these. You do need to know their names and which ones match each other. So cytosine and guanine will bind to each other. So if you see the yellow and the green, they bind to each other always. You never will have a cytosine binding with a thymine or an adenine. It only binds with guanine. Adenine and thymine will bind together. Okay, so you will always see the red and the blue together. Now that complementary base pairing is very important because then it allows for them to hook up back with each other and creates this way that we can actually make copies of DNA because of this complementary base pair. So if you open up the DNA strands, you're going to have an A, and if it comes into contact with a T, they're going to want to bind each other. So you can actually create new DNA because of this complementary base pairing. This allows for DNA to act as the molecule of inheritance for the body and for living things. Okay, so inside of each of our cells, we're going to have DNA, which allows for that, the instructions for us, what makes us who we are, that is going to be encoded in there. And we can also pass this DNA on to our next generation through reproduction, and we'll talk about that specifically. Now, RNA is going to be a little bit different from DNA. So RNA is going to have ribose sugar instead of the deoxy. It's also going to be single-stranded. So you see that we only have 1 strand right here. It has a 3rd change that makes it different, and you're going to notice that we have cytosine, guanine, adenine, but all of a sudden we have this new base called uracil, and it's not thymine. Okay, so in RNA you do not have thymine, so there should never be a T in your RNA anywhere. The T is replaced with uracil. Now, the reason why there's these slight changes between DNA and RNA is because your cell machinery needs to be able to tell the difference between DNA and RNA. DNA is going to be stuck inside the nucleus, and it should not ever leave. Okay, the nucleus is kind of like this very locked down, safe place that is going to hold those directions, because if those directions get messed up, you're going to have a lot of problems. But the RNA needs to be able to move throughout the cell so it can go make proteins. And that is going to be able to move because it has these special uracil, and it's also single-stranded, and it has that ribose sugar. So different machinery within the cell is going to be able to recognize this, and it's going to be allowed to move through the cell in a way that the DNA is not allowed to move. So here's kind of an overall picture of DNA. You can see the sugar phosphate backbone. So the sugar, if DNA, this is going to be deoxyribose, you're going to have a phosphate and then a sugar, a phosphate and a sugar, a phosphate and a sugar. So the backbone, this blue structure, is going to be this phosphate sugar backbone. The base is going to be attached to the sugar pointing directly inside, which is where the bases are. Okay, we have adenine, thymine, guanine, and cytosine. Adenine and thymine are going to be complementary, so they will always bind to each other, and guanine and cytosine will always bind to each other. Okay, this shows a little bit about how the two groups pair. When you have adenine and thymine, they will bond twice with these hydrogen bonds, and guanine and cytosine will actually have 3 hydrogen bonds that connect them. Okay, and we will then talk about this next part from DNA to proteins in a future lecture. Okay, so we'll talk about how we take DNA, turn it into RNA, and then make some protein from it. Okay, and you're going to have that RNA sequence that is going to be translated into the amino acids that we just talked about for the proteins. So I wanted to end this lecture talking to you a little bit about lactase and lactose. And many of you probably are lactose intolerant, and that means that you are no longer able to digest milk sugar. So milk sugar is called lactose. So I want to explain a little bit about how this works. So if you're a mammal, and we are mammals, you are born and your mother is most likely going to be feeding you milk that she makes. Okay, and inside the milk you're going to have a sugar which is called lactose. Now lactose is a combination of glucose and galactose. It's those two monomers or monosaccharides that have been joined together to form a disaccharide, which is called lactose. Now your body can't actually use lactose when it's has the two sugars in it. It has to break it down into glucose and galactose so that you can actually use those sugars. There is an enzyme or a protein called lactase. So this ASE is going to tell you that this is an enzyme. And I'm going to talk about enzymes, but enzymes are proteins that help chemical reactions take place. So lactase's job is to take lactose, the sugar, break it down into glucose and galactose. And when we are babies, we make plenty of lactose, our lactase, to break down that lactose because we're eating it. However, if you look at the life history of most mammals, after an animal is weaned, which means that it's no longer drinking breast milk, it will no longer come into contact with dairy. It just, in normal situations, there is no source of dairy that they can eat. And so they stop making lactase because they're no longer going to be eating anything that has lactose in it. Why would you make this protein? Because it's expensive to make it. Why would you make it if you don't need it? However, there are many humans that can actually digest lactose in adulthood. And this all has to do with a mutation that took place over 10,000 years ago that gave certain individuals kind of an advantage over those who couldn't digest lactose throughout life, because they were then able to utilize another resource, which is dairy products. Okay, so lactase is a protein. It is the enzyme that breaks down lactose, that milk sugar, into glucose and galactose. If you are lactose intolerant, you are not able to break down lactose in adulthood, and that's normal. That's a pretty standard situation. So if you have a dog or a cat, they normally say do not give them milk or any dairy products after they're a puppy or a kitten, because they won't be able to break it down and you're most likely are going to give them diarrhea or horrible digestive issues like farts. So that's pretty standard in most mammals. However, many humans, including myself, luckily, can digest lactose throughout adulthood and not have any digestional issues because we still produce lactase. So what they wanted to look at is, why are there certain groups of individuals on the planet that can't digest lactose in adulthood? And so if you look, I'm going to show you a map and it'll show you different regions, but certain areas like Europe and parts of Africa, specifically East Africa, have very high percentages of individuals that can break down lactose in adulthood. But if you look at other areas like Asia, they are not able to break down lactose in adulthood as much, and they tend to be at much higher levels lactose intolerant. So what they did is they looked for a genetic change in the genes somewhere near the lactase gene. So they found a single nucleotide change, so it had one of those ATGCs was changed, and it was 14,000 bases away from the lactase gene. And it turned out that in the individuals that they tested, which were 196 Finnish lactose intolerant individuals, every single one of them had that base change. So something about that specific base is going to prevent these individuals from making lactase. So they can no longer produce they can't make it, and then they can't digest lactose. And if you are lactose intolerant and you eat dairy, you usually are going to have pretty bad digestive issues. So some of it's not too bad, but if you're really lactose intolerant, you can be in extreme pain. So when they looked at these individuals and they saw that if you kind of look at the DNA, you can find on one of the chromosomes this lactase gene, and 14,000 nucleotides away, so that's 14,000 ATGCs away from it, there's a single nucleotide that has been changed. And if you look at people that are lactose intolerant and people that aren't, you're going to see that they have different nucleotides at those two sites. Okay, now it turns out that there are hot spots that individuals that are lactose tolerant live. Okay, and so if you look at this map, you see that if you are on the lighter side, so white and these pale blues, you're going to be in areas where most people are lactose intolerant, so they're not able to digest lactose. You can see other places where almost 100% of the individuals are lactose tolerant. So what happens is that in these areas where we have this very dark blue, those individuals had a mutation that took place that allowed them to produce lactase in adulthood. Okay, and it allowed them to utilize different resources like cow's milk. Okay, if you look at individuals that are lactose intolerant, so most African Americans are going to be lactose intolerant because most of Africa is also places where there are not going to have lactose tolerant people. Most of Asians Americans are also going to be lactose intolerant, but if you're from Northern Europe or Western Europe, you're going to tend to be tolerant of lactose. Now, the interesting thing about this is that this is going to connect back to the cultivation and domestication of cows. So if you look at Europe and Africa, specifically East Africa, there were certain groups of individuals that started to domesticate cows around 10,000 years ago. And this is when we think that that mutation took place independently. So the ethnic groups of East Africa and the groups in Europe were different from each other. They were not breeding with each other. They were completely separate, but both of them had that same genetic mutation that took place. So in these two regions, individuals that were able to digest lactose in adulthood were healthier, and they were able to have more babies, and therefore this mutation actually spread throughout these different populations. And so that's why you have two groups kind of in the European area and East African area that are able to permanently make lactase. Now, one thing that makes this connection is that we can look at cave drawings in these different areas, and you can see kind of at this time, about 9,000 years ago in Europe and 7,000 years ago in East Africa, they started to do drawings of cattle. And so we think that they were starting to domesticate different types of livestock, sometimes goats and sheep. And they would actually start having access to these dairy products. And that's why we think that mutation was beneficial. So the mutation was purely a coincidence. It was, it didn't happen because there all of a sudden was milk. It's just there's a random chance that you can have something like that happen, and it happened to work well in these situations because they had access to these different dairy products. Okay, so this allowed for a survival advantage. The climate in these areas, specifically in Europe, it would be very cold in certain parts of the year, and so you couldn't necessarily rely on different crops. And so if you had cows and the dairy, you could then use that as a food source. Okay, so very interesting evolution connection between when the cattle were domesticated and when people started becoming lactose tolerant through adulthood. And here are just a couple more of these images. These are thousands of years old, but the significance of our ancestors painting these on the wall shows that cows and other type of livestock were becoming very important to their lifestyles. This is a final kind of table that summarizes a lot of what we just talked about. It goes over the large biological molecules, the functions, and the different components of them and examples. And so today's lecture, if there's anything that was really confusing to you, please reach out and email me and I can go over it. We're also going to go over all of this in lab and kind of break it down and look at the different biological molecules, but it's really important for you to understand these things because they're going to keep on coming back up in future lectures. All right, so I'm going to end there. Thank you.